A little bit about the M-Cube that I worked with. I worked with professors from LS&A Natural Sciences, the graduate program Cs, and Civil and Environmental Engineering. So the Upper Mazaruni River is a unique aquatic ecosystem that's located in the Guyana Shield, which is a region of South America. What makes the Upper Mazaruni River so unique is the fact that many of the species there are endemic, which means that they only exist in the Upper Mazaruni River. And right now, they are being threatened by human impacts like gold mining, for example. And any reduction in the diversity of this fish community could lead to their extinction. Oh no, come on. So the Upper Mazaruni is full of underwater structures like rocks, rapids, and waterfalls. Now, changes in underwater structures can alter the distribution of the fish communities at that habitat. Right now, gold mining is altering the habitat structures of the Guyana Shield. During the gold mining process, deep substrate is dredged up, which both increases the turbidity of the water and forms these sediment mounds called tailings beaches. Throughout this process, many of the microhabitats in the area are lost, and this makes it unsuitable for some fishes. So morphology is the study of the form of something, in our case, the form of the fish. Fish body shape and fin shape and other morphological features dictate which habitat they may occupy. For example, deep-bodied fish tend to occupy still, slow-moving pools, whereas elongate fish tend to occupy more complex microhabitats like rapids and rocky substrate. Because of this connection between morphology and habitat, it's likely that the gold mining in the Guyana Shield will reduce the diversity of the fish community. I'm using fish specimens collected in the Upper Mazaruni to answer the question, is gold mining reducing the fish diversity? So the specimens were collected by my M-Cube team in the Upper Mazaruni River from both mined and pristine sites. Now, before I could start collecting data, the collection needed to be cataloged at the Research Museum Center. So jars were organized by both species and the field site that they came from, and I went through and counted how many individuals were in each jar and measured the largest and the smallest. And then finally, I collected the environmental and location data and using different spreadsheets, related it back to the specimens for the collection. Now, museum collections are a very valuable tool for evolutionary biologists and ecologists because they allow them to take an in-depth look at fish anatomy and to compare modern-day specimens to specimens from 100 or 150 years ago. So I chose 5 to 10 of the largest individuals of each species, and I measured, weighed, and finally photographed them. I took 3 photos of each fish, one from each of their unpaired fins, so the dorsal fin, caudal fin, and the anal fin. These 3 photos were then overlaid in Photoshop, and lines were drawn through the strings to find the center of mass. Then this photo was moved to a different photo editing software called ImageJ, which allows you to take measurements from a photograph given a set scale, like the graph paper that's behind the fish. From there, we measured the moment arms. A moment arm is an expression of the fish's ability to turn given the positioning of its fins. So we measured the moment arms from the middle of the base of each of its 5 fins. Then after that, from just the dorsal hanging photo, we took 8 different linear measurements, again using ImageJ, and both the moment arms and the linear measurements are related back to swimming function in previous literature. So, using all 13 of those variables, we created a principal components analysis. Now, a principal components analysis, or a PCA, is a tool that we use to identify which of those 13 variables account for the most variation between species in this community, and also to visualize that variation. So here we have on the horizontal axis, which accounts for the most variation, we have body depth, and that body depth is changing, so it's increasing as you move to the left, which you can see in the photographs of the fish. And then on the vertical axis, we have the variables that account for the second most variation. And at the top, we have longer anal fins, and as you move towards the bottom, we have longer dorsal fins. Now, we also included relative abundance data, which we represented by the circles. Yellow circles are mined sites, and blue circles are pristine sites. So the size of the circles indicates the relative abundance at that type of site. Where the yellow circle is larger, that means that that species is more abundant at mined sites. And where the blue circle is larger, that means that that species is more abundant at pristine sites. Overall, we found that 10 species were more abundant at mined sites and 14 species were more abundant at pristine sites. And we noticed that at the mined sites, those species tended to have more generalized features, like, for example, the tetras, which is a group of fish, at the top. Now, their generalized features allow them to survive and thrive in a variety of different habitats, including the mined sites, whereas some of the more specialized fish, like the catfish that are at the bottom right corner, are usually less abundant in mined sites or sometimes not present at all. And this is because these catfish are very elongate. So they're more specialized for places like rocky substrate and rapids, and those are the exact type of environments that are being destroyed by the mining. So overall, this PCA suggests to us that the community of fish at mined sites is likely a simplified one, and if mining continues to expand throughout the Upper Mazaruni River, then the community of fish will simplify as well. So in terms of future research, we'd like to add more variables to our data analysis, specifically from different kinds of photographs, like the lateral photograph that you can see here. And we'd also like to include some more of the environmental data that Dr. Kotel collected for us to determine how exactly mining is affecting the physical aspects of the river. So my M-Cubed experience has been a very rewarding one. Throughout the past 10 weeks, I've learned valuable museum collection skills, like how to handle preserved specimens properly, how to catalog a collection, the basics of fish anatomy, as well as many computer program skills like Excel, Photoshop, ImageJ, and the basics of the coding language R. So I'd like to take this time to thank my mentors. First of all, you guys have been the perfect example of a mentor throughout this entire process, and I'm very thankful for that. And I'd also like to thank the M-Cubed Scholars Program. I've grown both personally and professionally throughout this program, and I'm extremely grateful for the opportunity to present my hard work to all of you, so thank you for listening. Does anybody have any questions?
I was wondering if there is any historical data on the mining sites. It just seems like correlating the biodiversity with the onset of mining might be difficult, as opposed to maybe mining sites being selected in less biodiverse areas.
Well, so I think we only have data going back roughly 10 years. That's when my mentor started going there, and by that time the mining had already started to occur. So we don't really have a baseline data for that. Okay. Thank you.
Who's going to unhook you?